Hi everyone, I am Gursimrat Singh and today I've got this topic chemistry bioenergetics for a project and we're gonna talk about it in this video. Here are all the topics that I have got to talk about. First of all, we are gonna talk about some of the background information about biogenetics, some of the subtopics, some of the scientific discoveries that have led and some of their advantages and disadvantages. First of all, we are going to talk about what bioenergetics really means. So, it means the study of transformation on energy in living organisms. It helps us in understanding how living organisms acquire and transform energy to do the work. We need energy in our daily lives to perform every simple work to every complex work. In the form of energy, living organisms make chemical bonds and break them, which releases the energy. Humans require energy for many different tasks, such as the mechanical works and the chemical synthesis, as well as the anabolic growth that our body goes through. This energy is released with the breaking of a smaller bond and making of a stronger bond, which releases reusable energy. For living beings, ATPs is the energy currency in our body. All the metabolic processes help us in synthesis of ATPs, which stand for adenosine triphosphate, and gets broken down into ADP, stands for adenosine diphosphate. The ratio of these ATPs to ADPs in a cell is called its energy chart, which helps the cell to relay information about its energy needs. The process used by cells to produce ATP is called as oxidative phosphorylation. Wherever the ATP is more than ADP, the cells use ATP. Whenever the ATP count is less than the ADP count, there is a need for cell to generate more ATP using the above mentioned process. Now we are going to read about some of the types of bioenergetic reactions. First of all, we're going to talk about exergonic reactions. Exergonic reactions refer to the chemical reactions which release energy in the form of heat, light, or other molecules. These reactions can occur in a closed system to retain a temperature or pressure. In case of living organisms, exergonic reactions are catabolic in nature, where some macromolecular substances are broken down into small molecules of different substances. For instance, starch and glycogen are broken down into glucose to produce energy in our body. Secondly, we're going to talk about endergonic reactions. In opposition to the exergonic reactions, endergonic reactions are processes that consume energy. This sort of reaction will not occur in a stable temperature condition and in a closed system unless a specific amount of energy is provided to it. In a living organism, these reactions are anabolic in nature. In anabolism, the energy is released from catabolic reactions, supplies the required energy for the synthesis of biomolecules. Now we are going to look forward to the some of the scientific achievements that we have made due to bioenergetics. In the past, it was usual for researchers to disrupt a tissue and isolate the mitochondria in order to investigate the role of mitochondria, which used to be really hard and time taking. But now, researchers have found many different techniques that allow the researchers to investigate the bioenergetics of a cell a lot easier, as well as with better precision. This new type of method is called the cellular bioenergetics. Cellular bioenergetics has advantages as related to normal classical studies using isolated mitochondria, but it also has its limitations. Some of the limitations are that many reagents can be used with an isolated mitochondria and cannot be used with an intact cell, which might cause different problems. There is research in bioenergetics has also led to autophagy, which is, which is helpful in treating cancer. Now we are going to look at some of the advantages of bioenergetics. With increasing research in bioenergetics, we have learned about autophagy, as I talked about it, which means self-eating in Greek and which helps in cancer therapy. Autophagy is a very important process in humans that pathogens, proteins, or organelles are thrown or waste or reused for anabolic needs of cell. Autophagy can be regulated by several different factors such as stress, including hypoxia, nutrient starvation, ATP and AMP levels, and any microbial infections. Autophagy triggers the cell death mechanism, which helps in removal of any cancerous cells. To mitigate the cytotoxic effects of cancer therapies, autophagy is often, if not uniformly, activated to remove protein aggregates and nuclear damage. We are now going to talk about the advantages of cellular bioenergetics, which has many advantages as it is better for physiological aspects of the cell organelle we are going to test, as they are left untouched and good for testing more precise outcomes. This method does not require the isolation of any type of organelle from the cell, which leaves it as natural as it can be. Saves money and time of the researchers, as the simpler way requires the experimenter to isolate the mitochondria from the cell, which is difficult or impossible to do in any animal, and cell lines from tumor can build up in this procedure if not done in the right way. Here are also some disadvantages that we have got to know about after we have researched about bioenergetics. First of all, bioenergetics processes are essential for cell performance. Deregulation of these processes has been studied to associated with different severe problems such as cancer, diabetes, and other neurological diseases. Several different connections were seen between metabolic defects and cancer epigenetics. Cancer is either genetic or epigenetic. Epigenetic enzymes depend on ATPs, metabolic and dietary factors. Hence, bioenergetics provides an important interface between the environment and the epigenome. Here is the list of all the citations that I have gathered my information from. Thank you everyone for tuning in and hanging till the end. Hope you all must have liked my presentation and have got some information related to bioenergetics. And thank you.